Okay, now I will soon ask the standard question. Can you see my screen?
Yes.
Yes, good. Okay, so today's topic, that is marine geology. So I have 2 questions to begin with. What is the topic for today's marine geology, marine micropaleontology, oceanography or paleoceanography, or yeah. And this is my cat, Mone. Mone. And who am I? I am Helena Filipsson, as you know. I am a marine geoscientist or marine scientinista. And I am in particular interested in areas that are experiencing low oxygen in the bottom water, like the Baltic Sea, but there are also many other places. So first of all, I thought I will introduce myself a little bit so that you know who is talking, and then we will go into the marine geology aspects of this. So marine scientinista. So this is my background. I did my master and my PhD in oceanography at the University of Gothenburg. And then I went on a postdoc to Bremen for 2 years. Then I moved across the pond over to Hui, to Woods Hole Oceanographic. They are the ones who found the Titanic, for instance. So I was there as a Fulbright scholar in 2006. And when I came back from the US, I moved to Lund. And then when I met Anna as well, and Kalle. And I also moved to Älmhult, the IKEA town. And since then, I have been in Lund, and I'm a full professor there since 2016. But I have also lived in Japan, in France, and in Gran Canaria. And I tend to bring my family along when I can. So here we are in Japan. My husband was on parental leave at the time, and we were there for 2 months. And here I am out at sea with my students. Over here, up here, you see a picture of me out on the fjords when I'm a young PhD student. And here I am, basically on the same fjords, also a low oxygen area, working with my students. And I have a favorite organism. I asked you about your sort of tool of practice, and mine is foraminifera, or forams. And after this lecture, you will all be more or less experts in the field of forams. And there will be an exam in the end, of course. So that's me. And we live in Älmhult in the IKEA town because my husband works for IKEA. That is more or less the only reason to live here. So, as I said, I'm a marine geologist. I get the opportunity to go out at sea at big research vessels, like this is the Ocean Surveyor from the Swedish Geological Survey. And what I do is that we take up long sediment cores or short sediment cores, and we use them as environmental archives, basically similar to what Anna and Kalle are doing on lakes. We are doing out at sea instead. And we use them as environmental archives. So here we have a core from Greenland, and here we have one from the Baltic Sea. And here you see a picture from the large archive or storage facility we have in Bremen, where we have cores from all over the world stored in these large containers, or not containers, cold rooms, basically. So what do we do with these? And this is, you know, we have our sediment cores, and I say that they are environmental archives, but we need to read these archives. We need to understand them. And to do that, we look at lots of different tools. It could, for instance, be laminated sediments. That's what you see here, with the banding in the sediments. And this banding is preserved when there is low oxygen environment. So we know that when we have had low oxygen conditions, then we also have these laminated sediments. So that is one indicator we can use. We can use also foraminifera, and as I said, I will explain in detail what this is, but these are all different kind of forams or foraminifera. And we also, other tools we're using, that is a much smaller organism, that is coccolithophorid, or coccolithophores. And I will show an image of this in just a minute. So please note this little coccolith, that is the plates that's forming the coccolithophorid. And then we can also use dinoflagellates. That's the picture to the down in the lowermost right corner. And we can use also not only what kind of species it is, but also the geochemical composition of the shell. Now I have to ask you, do you see my pointer? You see when I'm, okay, great. So that is one of the some of the tools we can use. And as I said, I'm very into forams or foraminifera. But these are all on the micro scale. And I said earlier on that I was a marine micropaleontologist, and just to illustrate the micro part of this, you know, paleontology is when you're working with dinosaurs. Micropaleontology is working with tiny things, but how tiny are they? So if we're just looking at the eye of a needle, for instance, and we put a little planktonic foram in here, and this is a planktonic foram, it's living up in the water column. And then we're zooming in on this. And you see here we have an opening in the shell. And if we continue to zoom in here, you see that this particular foram is forming little spines on its shell. And next to that are also coccoliths. And that is just a coincidence that there's coccoliths that has been attached to this foram shell. But there you also see how incredibly tiny this is. So if you go from the eye of the needle down to the coccolith, you can understand what kind of structures that we are looking at and how small they actually are. And these are the ones that we use to understand the past marine environment. So coccoliths, you ask, so why do we care about them? Well, they are, for instance, that is what makes up the White Cliffs of Dover. Basically, it's coccolithophorids to a very large extent when you see white chalk cliffs like the ones at Dover or out on Møns Klint here in Scandinavia. And so despite that they're being so small, being enough of them under a very long time, then they will also be an important part in the bedrock. You also see here the very bluish color here. That's a bloom of coccolithophorids. This is not a poisonous bloom. We have algal blooms that is not very nice at all, but this is not any toxic one at all. It's just that it's a lot of coccoliths or coccolithophorids that are blooming at the same time. So despite they are so tiny, if there's enough of them, they can form blooms that we can see on satellite images. And these actually, they also create a small substance that can act as a cloud formation nuclei. So these blooms can also affect local weather, or in further perspective, also influence climate. So despite being so tiny, it can have a huge impact. So this is the coccolithophorids. Yes, Josephine, I think you have a question.
Yes, I do. Are they an organism, or how would you?
So coccolithophorids are phytoplankton. They photosynthesize. So they are in the surface water, and they are using the sun for energy and producing organic matter. And they are very important in the carbon pump for these aspects. They are both producing organic matter, and they also produce calcium carbonate through their skeleton. And then the organic matter, when they die, will sink down to the seafloor. So they are very important in the carbon cycle as well.
Can I ask something?
Yes.
This photo from above, is that only the camera that sees this blue, or can the human see it?
We can see it too. We can see, but I think this is a bit digital enhanced, but we could see it too. Yeah.
Okay.
No, I don't see all of you. So right now, I just by chance that I saw that you are waving. So if you have a question and I don't recognize it, just speak up a little bit. So then I because I only see 4 people at the same time. All right. So basically, and I love this quote, that these tiny organisms and the tiny shells, they are really the true heroes for our quest to unravel the past of our planet. It's a colleague of mine who said this, and I think that it's really so true that these tiny microorganisms, without them even knowing it, that they are the superheroes. So forams, as I said, you will all be experts in foraminifera after this lecture. So forams foraminifera, or forams for short, they are single-celled organisms. And I think that is just that fact is rather impressive. They are a single-celled organism, but they can have so many different shapes and forms. And you see pictures there in the lower part of the screen. That's all different kind of forams. They typically have a shell. They don't have to have one, but they typically have, and we are mostly interested in the ones that has a shell because then they are preserved in the sediments. And they have been around for a very long time. And the size is typically a size of a sand grain. So you need a microscope to see them basically. And they are so abundant. If you have been at a beach, which was a little bit muddy, like a tidal flat or so on, you probably had forams between your toes. If you were swimming in the open ocean, you probably swam among forams. They are very, very abundant. And together with coccolithophores, they form the major part of calcium carbonate. So, and they can be planktic. That means they live up in the water column, or they can live down in or at the seafloor within the sediments. And there exist around 50 planktonic species and around 4,000 benthic ones. So in the benthic, there's such a large different kinds of environments that you can live in. So that's why we have so many different kinds of species. And the shell, we are mostly interested in the ones that have a shell made of calcium carbonate, but there are also shells that are made from sand grains. And you see how down here, there's one that's picked up different mineralogic particles, little sand particles, and build a shell out of that. And they can be extremely picky. They can be that they are only picking fragments of a certain size. And I think this is just fascinating. If you are a single-celled organism, but still you have very sort of clear idea what you want to build your shell of.
So,
and let me finish. And then also, so forams, you can classify them as zooplankton. They can eat other forams. They can eat organic matter. They eat phytoplankton, so coccoliths, for instance, or they can also hunt for bigger prey like shrimps. But they are protists. They are single-celled organisms. That means that they are not an animal. They are not a plant. I usually say that they are single-celled organisms with traits as an animal. They are sort of animal-like, but they are not an animal. Okay, Josefin, you had a question.
I have a new question now. What makes them animal-like? What's the...
It's just that they are hunting for prey. They also, I don't have a picture of that, but here now, but they have like a a structure around them that is like a spider web, similar to spider web. It's called a pseudopodia, and that stretches out around them completely, and they use that to drag themselves around with it, and they also use that to catch prey, and they use it to get rid of products they don't want. So they have sort of many, they are heterotrophs, they're eating other organisms. They have, they can both have asexual and sexual reproduction, depending on what kind of environment they are living in. So that makes them similar to an animal, that's right, but they're not animals.
So pseudopodia, does that sort of translate to a fake foot?
Yeah, that's true. Yeah, yeah, but you can think about it as that they can stretch out a spider web around them.
And does this come
in all different directions?
Is this a product of their body, or is it something they make? You know,
it's a part of their sort of body, of their cell, extension of their cell. I will let me later on. I will pull out a picture showing. So, but basically, think about it as a three-dimensional spider web around them. But this is not preserved in the sediments, so we can't look at it when we're going back in time. But we can see this in living forams.
Can I ask something?
Of course.
Okay, and I have my camera off because my phone, so that you know. What is the name of a species? What is in between and an animal? Because you said it and I missed it.
You say what categorized
like,
it?
The foramin is a species between. Yeah, you named it in a certain way. Like you said, you have an animal and there's a plant, but this is.
But this is a single-celled organism.
An
a
animal.
Protist. It's a protist. Was that the word you were looking for?
Protist, maybe. Yeah, I think it's another word, but otherwise I will listen in the recording and then I will write down the word protist. Yeah.
Yeah.
Thank you.
Yeah, no worries.
Is that a protest with an I? Protist.
Protist.
Okay. Protist, huh?
Yeah.
Yeah.
Protista. Okay.
Okay. Yeah.
Yeah, great. So here we have some more pictures of forams, and you see that they are so pretty. But what we are really interested in is not only what kind of species that is living there, when that's what we are using as well. What kind of species do we find at certain locations? But we are also very interested in the geochemical composition of their shell. So first, this comes first. So forams, this is a silly cartoon, but in some ways it's rather useful to show how forams are, what is limiting their distribution. Of course, it is both salinity and temperature, especially planktonic forams. They can't live when the salinity is too low. But here in this little cartoon here, you see that you have planktonic forams up here. They're eating coccoliths, they are eating diatoms. They will fall down to the seafloor. And you will also see that the forams, depending on which species it is, will live in different sediment depths. So certain species are adapted to live at certain depths in the sediments, depending on what kind of chemical gradients that are present in sediments, and if they are adapted to live in low oxygen environments, or if they are adapted to live with the strong bottom currents, for instance. And you see that we have some species that actually prefers to live attached to a little rock or attached to a seagrass, for instance. So as I said, we are very much interested in the geochemical composition of these shells that the forams are precipitating. And the study of forams or foraminifera goes back a long time. So these are figures or Christmas cards, I would say, that two researchers sent to each other in the early 1900s. You see here, they have made the Christmas cards from forams. So they've been working under the microscope. And then you see here is Arthur Earland. He sent a Christmas card to his colleague, Edward, and from 1912. So they were doing this, and they were using forams from the Challenger expedition, which was in 1872 to 1876. And this expedition is sort of called the birth of oceanography in many cases. So this collection of forams is still available at the Natural History Museum in the UK, or in London. And later on, they had a fallout and they became not enemies, but not very good friends any longer. But it's a very nice Christmas cards they were sending to each other.
Can I ask something?
Yeah.
I just, now when we look at it, it looks like tiny shells and stuff that has been like picked up. That makes me think that it's not as tiny as a sand grain. Or how to know that it's a foram and not as, like, can you really differentiate, or can they also be a little bigger than grain size?
So yeah, so when you see if you would put this next to a shell, like a mussel shell, then these would be enormously big. And even when so this is all done, I think you have to understand, this is all done under the microscope. So this is not absolutely not something that you can touch with your hand. So it's very impressive that they did this and glued the forams into these Christmas cards. These are tiny, tiny, tiny objects. So if you think about them, this is a quite big one, but this would be basically two quartz particles next to each other, two sand grains next to each other. So it is tiny. And you can see juvenile mussels, for instance, in my samples, but they are always much, much bigger than a foram. So this is why this illustration with the needle as well. If you think about the eye of a needle, the size of that, and then you remember the foram, that is the size of these. And then you have some long elongated shapes, but these are all tiny, basically. But I agree with you. They look like shells that you find on the beach. And you will find these on the beach. It's just that you need a microscope and see them. And yes, this is another quote that I really like as well. And now I've been talking a bit about the shape of the forams and how pretty they are and how we use them, just looking at what species we find and how we use that to reconstruct past oxygen concentration or past how much food there was available for them. But as I said, we are also very much interested in the geochemical composition of it. And that is because, so this was, you saw the Christmas cards, that was like 1912 or early 1900s. Then in the 1950s and 60s, mass spectrometry was invented, and people realized that, wait a minute, we can actually analyze these forams, and we can see that the concentration or the ratio between oxygen 18 and oxygen 16. So oxygen 16 is the most abundant form of oxygen we have. But then we also have oxygen 18 and oxygen 17. And you can measure the ratio between these. And they noted that the ratio between these isotopes varies depending how much ice is available. If we have an ice age and lots of ice is stored up on the ice caps, we will have a much higher delta 18O value in the oceans compared to when we are like we have today, when we are in an interglacial. And they realized that you can go basically sampling all across the world. So these are sediment cores from all across, the Pacific, in the Atlantic, and analyze the benthic foraminifera and see the same pattern that we would have. This is present day, and then it goes 600,000 years back in time. And you see the same pattern going from interglacial to glacial. This is the glacial. This is the last glacial maximum. And you can see the same pattern everywhere. And you can use this to correlate between different sediment cores. And this was a major, major revolution when this happened. And when people understood how it's all connected and how it's connected with the glacial and interglacial timescales. And here you can see the forams, they are sitting there. It's not like they can't really decide. They sort of passively record the temperatures and the environment around them.
Can I ask a question about that? What does that mean, to passively record?
So that means they can't actually, to some degree they can choose, but to a larger degree, they just, depending on what temperature it is, they will take up more 16 compared to 18, or what kind of water that's surrounding them will determine what ratio of 16 and 18 is built in their test. It's not like they are sitting there saying, no, we don't want that much 16 of oxygen. It's more that they build what is around them.
So there wouldn't be another situation where they could actively record it.
They can sort of do it, especially when they're young. It's a slight difference between what is in the water and what they actually are recording. But overall, they are just picking it up what's around them, which is great, because then we can use them as thermometers going back in time. So then now we have talked about how we use them, what kind of species we find and how they look, the morphology. And then I've talked about the isotopic composition of their shell and how we can use that, sort of the geochemical composition of them. And then now we're combining those things, and instead of just looking at them using ordinary microscope, we can also look at them through synchrotron light. And MaxLab is on the picture here. That's a synchrotron facility that is built in Lund, and you will find these synchrotron facilities all over the world. It's like a big ring. It's the size of Colosseum almost, and where you're charging electrons. And then you're bending the electrons. They're forcing them to bend, go around in a circle through big magnets. And then when they are sort of charged, they will release a photon, so light, basically. And then you can use that light at certain energy levels to and hit that beam of light through your sample. And then you can analyze all sorts of different objects. For instance, we use it to look at forams, but you can also do that with paintings. I think you have all seen that. You can use that to see look at layers of paint, and you can basically see another picture underneath the painting. There you can do with synchrotron light. And you can also analyze all sorts of material and objects. But we use it for look at forams. So we have this very, very, very big facility to look at the tiny, tiny forams. And you can basically think about it as one very big microscope. And we do micro XRF, and that means that we are making maps of elements in the foram shell. And we are doing micro CT scanning. That is, we do 3D dimensional X-ray images of the shell. You can, if you go to the hospital, you can do a CT scan of your brain if there is some sort of different kinds of tomographic investigations. You can do that also with on forams, but the big advantage when you're doing it with synchrotron light is that it's much, much quicker. You wouldn't do that with a human, though. You wouldn't put synchrotron light on a human person. So this is what it can look like. I apologize that the images are not perfectly attached here, but you see sort of So this is a forum at Max Lab in Lund. And here we are looking at both the calcium carbonates, that's the picture to the left. And then we're also looking at the distribution of manganese. In this project, we were interested in the distribution of manganese in forams. So I think that it looks pretty. And here it's pretty nice. And here it's really interesting. You see, you have seen how tiny they are. And then you can use the synchrotron light to look at the element distribution in the shell wall of these of the forams. It's pretty spectacular.
I have a quick question.
Yeah.
So like the left and the right photo, you see the shell and then what's inside of the shell to the right, right?
Well,
Correct?
you see in this picture, you see actually they are both what you see inside, but it's two different elements. The one to the left is the calcium carbonate, sort of, and then to the right you see the distribution of manganese, just that element within the calcite.
So it's a single cell organism and there's like something in, like there's a shell and then there's something inside of the shell. So does the thing out inside of the shell like make its own shell?
No, it's elements. So, you know, if you would take your skeleton and you would analyze, no, I don't see you. I have to scan here so I see who I'm talking to. Yeah. So, you know, if you would take your skeleton and then you would analyze the composition of your skeleton, you would also have lots of different elements in there. It would be strontium, it would be calcium, it would be, yeah. And that is what we're seeing.
Okay.
But the thing is with the foram, it is that it's hollow inside, or it has the cytoplasma, the cell content, but that we have removed here. We have used bleach to get rid of that. So it's basically like, if you think about it as a shell you find on the beach, but a tiny micro one. You know, if you find one on the beach, it doesn't have any tissue inside, so it's empty. And then you would just analyze the calcium carbonate. Do you see?
Yeah.
Yeah. Great. Yeah.
Just to continue that question, so in a way, it's like the skeleton here is outside of the organism. It's containing.
Yeah, exactly. The organism is inside, but not here anymore.
And the organism grows that shell itself.
Yeah.
Even being a single cell, then it like kind of grows like a skeleton.
And it's so fascinating that it's a single-celled organism. It grows when it has decided that it needs another chamber. It can do that within a couple of hours. It grows another, and exactly how that is done is still debated, and still different sort of theories how it's actually doing it, and different species doing it slightly different ways. So it's a single-celled organism that is growing its own shell, and as you saw in the pictures earlier, the structure of that shell, how it looks, varies tremendously from different species. And as I said, it can also be a species that decides that, oh, I only want to build my shell out of that type of sand grain, and I find that on the seafloor, and then I pick it up and build my shell with that. And it's still, yes, a single-celled organism that is doing that.
So at some point when it's born without a shell, or it's
it's basically just a little organic lining, and then it builds tiny. The first shell is very small. Here is the first one, and then it grows. There's a second, third, and here is here you see this piece is a little bit broken, but this would be the youngest part, or the most recent addition.
So those different parts would be the chambers?
Yes. Sorry, I've completely forgot to say that. Yes, those would be the different chambers.
And so then, if I understand you right now, it's using parts of like organic material or inorganic material, but it doesn't grow the parts. It builds them. It constructs them from outside.
Oh, it's different from different species. This one does it by itself. It takes the elements it needs. It takes that out of the water column and builds its own test. So it has an organelle that is responsible for doing that. So it takes calcium, it takes carbonate, and builds calcium carbonate out of it. So it has its own process to do that. So it can both be a calcium carbonate shell or calcite shell that it has built itself, or it can pick particles from the seafloor to do it.
And does it then pass these, if it has different ingredients, or it's like a tiny chemist, it brings the materials together and then they become this amalgamation. But does it then do it through its body, or does it do it outside of its?
Oh, it has a calcification site of calcification. It's doing it in its body, and it can also regulate the pH when it's doing that. And it's all single-celled doing this thing. Yes, so through synchrotron methods, as I said, or we looked upon, we can make maps of how different elements are distributed within the foram shell. Here is a picture which I find very fascinating, because this is a foram from the Baltic. It has been living in an extreme low oxygen environment. And the green here is calcium carbonate. So that is the original shell. Then it has been, as I said, it's been anoxic, so extreme, no oxygen available, and very, very high amounts of manganese and iron has been available around it where it was deposited at the seafloor. So after it's died, there's been precipitation, sort of formation of iron inside the foram. So the foram was dead when this happened. So iron has entered the shell and forms this precipitate. So you see that in this particular foram, it had little pores, and you can see that it's more iron in the pores. Oops. And the red here is manganese that is covering the foram. And this is really an extreme example of a foram that is basically already from the beginning, it was on the limit where it could live. And then after deposition, after it's died and in the sediments, then all these other minerals have been deposited on the foram. So that was microXRF, where we are using synchrotron methods to look at the element distribution in the shells. Then we're also looking at morphology. So this is when we are doing the micro CT scanning. So basically we are doing 3D models of forams using X-ray. And this is Dirk, who I have been working with for a long time, and some of my students. They were over in Japan and scanned a whole lot of forams at the synchrotron facility there. And so basically they have to put a foram on a little stick, or we call it gecko tape. And then they put them in this in the machine. And this is called the hutch. And this is basically where the beam line will be opened and the light will hit the forams. Then we can get pictures like this, and I'm sure you have seen 3D images of lots of different kind of fossils where we are scanning using CT scanning. So basically here we see, but the big advantage when it comes to micro CT scanning is that we can slice the images in all sorts of different directions. And then we can look at shell thickness and shell weight and how they are looking, and if they are changing when the environment is changing. This is where what I am interested in. I'm not just interested in getting lots of nice pretty pictures, but also does the shell changes when the pH is changing, or the salinity is changing, or the oxygen, or is it always more or less constant? They can sort of overcome any sort of big environmental changes. So that is what we are looking at now, and what we are hopefully focusing on. We will have some bit more funding when we will continue this work to look at lots of forams from different time period, and how the morphology, the shape and the weight is changing. Here just using one example where we have forams from the Baltic, and we are going back the last 200 years to see if the weight is changing, or if the surface volume has changed, and so on. And here we are actually, we thought we were going to see that with ocean acidification that the mass was going down, but actually we can see that instead it's going up, and they are actually getting bigger. And the question is then, why is that? Is that because they are getting more food recently, or is there something else that is why they have become significantly larger over the last 20 years or so? So now we have gone sort of all the way from just looking at them with a microscope, observing the beauty of them, to then into the geochemistry aspect of them, and then going back to this very powerful microscope that the synchrotron light is doing. Finally, I just have a few other observations, and I have to make a confession here, and that is that I really love data. And in particular, I love hydrographic data, salinity, temperature, nutrient data, oxygen concentration data. I love that. Maybe that's why I studied oceanography. And I force my students to make plots like this always, where they are using environmental monitoring data to look at temperature, salinity, and oxygen concentration, how that varies. For this is throughout the year. And because I think that if you want to study the past, you really have to understand the present day. You really have to understand how is the salinity changing in this area, and how is the bottom water oxygenation changing over time. So when they make plots like this, it always makes me very happy. And if they don't, they have to go back and do it. Going back to data and hydrographic data, and how you view instrumental monitoring data. So then I want to give you an example that which we perhaps can discuss a little bit later as well. And that is, this will be examples from the Baltic Sea. So the Baltic is here. You know, we have the Skagerrak out here and the North Atlantic out there. And the Baltic is one of the most polluted marine areas in the world's oceans. And it suffers from large areas with very, very low oxygen content, or even anoxia, so total lack of oxygen. And that is illustrated here. Here you see time on this axis, and then you see area of, the red is low oxygen, the black is sort of dead zones. And you see how that has dramatically increased over time. Here's another figure that shows the intensity of inflow. So basically, if you want to have a water exchange into the Baltic, it has to come through Öresund here, or the Danish Strait. So here you see the number of inflow events and the frequency, how often they are occurring, and the total volume of them as well. And you saw that in 2015, we had one that's really, really strong, but then we have had very few over the remaining part of the last 5 years or so. That means that there will be very little oxygen, low oxygen levels in the deepest part of the Baltic Sea, and that the area is also increasing close to the coast with low oxygen.
Can I ask?
Yes.
Why was there such an increase in 2015?
This was because the weather. So the thing is, if there should be an exchange in a deep water exchange in the Baltic, the weather conditions have to be just right. So there has to be winds coming from the north to northeast that basically pushes all the surface water out of the Baltic. So basically the sea level is getting dropped in the Baltic. So it pushes it out, and then there is a change in wind direction, and there's strong westerly winds that will push saltier water with higher oxygen content into the Baltic. The problem is that if there's a very big exchange like this, that means that the salinity will be relatively high in large parts of the area. That means that if the exchange going to occur, then the salinity has to be even higher the next time. So we want to have lots of exchanges, but we don't want to have these super exchanges with comparably high salinity content. So just having westerly winds, strong westerly winds, doesn't make it. It has to be this sort of combination of first easterly or northeasterly winds that pushes it out, then the change in flow, and then it can enter.
And could this be connected to the overall weather situation in the world, or is it more local?
It's so we can see that when we have periods of when the North Atlantic Ocean, North Atlantic Oscillation, that's the sort of global weather pressure system with the low pressure over Iceland and the high pressure over the Azores. If we have a negative NO, that means that we have more winds from the north to northeast. That typically gives an advantage for an exchange. So it's all connected to the sort of global situation. But in the Baltic as well, we have to remember that it's not only the exchanges. Then we also have a very huge amount of nutrients coming into the Baltic that causes eutrophication and a very high oxygen demand.
I have one last question
Yeah.
regarding this, and it's different because all this terminology makes me think about the other talk of inflow in 2015, and I wonder, when you guys look at this so specifically, do you also sometimes see how there's a parallel to political events and just like inflow of, or exchanges?
Political events? Oh, you mean?
Just looking at the terminology of the flow of people of 2015.
I haven't thought about that, just because, yeah. But of course, yes, of course, it's also connected to. I think this specific event, I wouldn't connect that to 2013. I think maybe the big one here was even earlier, 2015. It was probably. Yeah, but yes, of course, it's connected when you think about areas where there's drought, for instance, or where we have El Niño years where we can experience drought in certain regions and excess precipitation in others. So another thing that is happening, it's not only that we have a lack of oxygen, we also then have an increase of hydrogen sulfide. That's sort of the next step on what happened in water chemistry. And hydrogen sulfide is the smell of rotten eggs, basically. And it's will come in the deep water when the oxygen is depleted. And here you see the concentration of hydrogen sulfide, and how that when we have when there hasn't been an exchange of deep water for some time, then you will see this build of hydrogen sulfide. And no high organism can live in that. Hydrogen sulfide is also very toxic for us. That is why we are so sensitive to it. We can smell it at extremely low concentrations. So that brings us up to the topic of dead zones. And dead zones is areas where you have anoxia or lack of oxygen. It doesn't mean that there is nothing there. Of course, we would have bacteria, and of course we will have different types of microorganisms. So it's not that it's completely dead, but any high organisms would quickly disappear. A fish, for instance, will not be in water with oxygen content less than 2 or even 3 milligrams per liter. It will swim away from that. And here you see a reconstruction of oxygen concentration in the deeper parts of the Baltic during time, and how that has evolved. And you see that there is expansions and contractions. We had a very large zone with hypoxia in the 70s. And then we had periods where the climate or the weather conditions were so that we had frequent water exchanges. And then the zone decreases. Whereas now, again, we are in a time where both with the combination of eutrophication, sort of pollutions from humans, and also how often we have had an exchange that has resulted in these massive areas with that we call dead zones. You see the black where it's completely void of oxygen. So what I think when I see this is that I have often thought about how can you present this in another way? As I said, I love data. But how can you make another interpretation to sort of get the message across, and how to perhaps visualize or how to interpret it, different environmental problems in different ways, sort of thinking about dead zones. In a map, I think it's quite sort of easy to get. But if you're looking at this graph or this graph, how can you explain this in another way? Can you sort of make music out of it? Can you show it in a way that people perhaps understand it in a different way? So that is what I want to leave you with. So I've shown you a lot of pictures of microfossils and marine records and isotope curves and salinity curves and so on. And I'm thinking, and I'm curious about how you are thinking or feeling about sort of interpreting or presenting the records, the environmental archives in another way, perhaps using just the data, shell weight, for instance, and show that beyond what I typically show in my presentations. So that's that. And thank you very much for your attention. Now I will stop sharing so I can see you. And I'm happy to take questions, of course.
I have a question. So I remember earlier on in the presentation, you said that the forams kind of record the temperature and the environment around them. Can you speak a little bit more about that?
Yes, so it is through their incorporation of isotopes and trace elements. So they will incorporate different amounts of oxygen 16 and 18, depending on the temperature when they are living there, and also how much ice that is stored out on the ice caps. But they will, when they build a shell, if it's relatively warmer, they will incorporate 16 just because it's easier to pick that up. And they will also pick up more magnesium and replace that instead of the calcite, depending on temperature. And this is also a process they are just doing without their deciding it on their own. So we have seen that there is a relationship between the magnesium-calcium ratio in the foram shell. So if we analyze the magnesium-calcium ratio in the forams, we can relate that to temperature. So it's sort of indirect. It's not like we can just look at the foram and say, oh, this was 4 degrees. No, this was 5 degrees. We have to analyze the elemental composition in different ways of the forams to relate that both to temperature and we're using other variables to measure salinity.
And just very basic, the forams build these shells as a survival strategy, like to protect themselves, or like, is it?
That's a very good question and a very interesting one, because there are forams that don't have a shell, but the majority do have a shell, or the ones we are interested in have a shell. But we also have forams in the Baltic where they have a shell, and then the pH getting so low, so the shell dissolves, and they are left without the shell, or they are just having a half shell, and they are still alive. And that makes you wonder, how long can they survive without a shell? But I think it is, it is a way to protect themselves. It is a way to protect the cytoplasma and to prevent being eaten up. Well, they are getting eaten by other organisms, but yes, for protection.
But so the reason why you're more interested in the ones with the shells is because the shell is the recording.
Yeah, the shell is preserved in the sediments, and we can record all this. We can get all this environmental data from them.
And so in your process, is it also important to follow the now living forams?
Yes, I'm very interested in that. And we are also doing experiments where we are culturing forams at sort of different temperatures or different salinities. It's quite complicated to do it and make them happy to grow in lab and so on. But it's a lot of fun. It's a lot of work, but it's a lot of fun to do that as well. That's what I did when I was at Woods Hole. I was culturing forams and very excited when we had babies.
So how big do these lab or what containers have to be for the?
Oh, they were tiny. So we had lots of them, but they were like this. And then there was a little culture cup that was maybe 1.5 centimeter in diameter. It was a lot of work. Just first we had to go out at sea and collect them, and then pick them up, the ones we thought were alive, and then put them in the culture cups and have them in controlled conditions over several months. So yes, that was a lot of fun.
I have another question about the process of this incorporating oxygen, how it happens.
Yeah, so basically they need, so it's the carbonate ion that they need together with the calcium, and it is what oxygen in the carbonate ion that's available in the seawater. So it's the oxygen there, if that is an 18 or 16.
So that part is sort of a byproduct of what they are actually doing.
They sort of they don't have a choice. Sort of they need it to build their shell, and then they take what's around them.
And why is it 18, 17, and 16?
Different elements have different isotopes.
Right.
Yes. So like carbon, for instance, the most of carbon is carbon-12. But we have also the radioactive carbon-14, and we have some 13 carbon as well. It's just the different amounts of neutrons that they have. So we have all sorts of different stable isotopes and then unstable isotopes. And oxygen can be in 3 different isotopes. Most of it is in 16. That's the majority. I think it's like 97% or something like that. I don't remember exactly. And then we have some that is in form of 18 and a very small amount of 17.
And they want 18 the most.
They don't want anything.
No, but they pick up 18 the most.
Well, they pick up more 16 when it's warmer, but it's just how the thermodynamics happens when they build their test. It's just that they don't have a preference. They can't choose. It just happens.
Right. And I would like to hear a little again about this facility. Like, I would like to hear, what did you say? You said that
Max Lab? No?
Yeah, like no. So this thing about, like, you know, what was it in these facilities? The synchrotron.
Yes.
Yeah, that magnets force what to, like, could you just explain again?
Yes. So I'm not a physicist, so you know, yeah. So what they do is that they charge electrons, so that they are, and then they're getting the electrons into this big circle, and the magnets. Then they have magnets on the side of the big ring, and the magnets are forcing the electrons to turn. And then when the electrons are turning, then they are losing energy, and then the photon will be released, like light. And this light will hit our object, like, and depending on the energy, we can analyze different elements within when the light is hitting the forearm or the painting or whatever. And then we can use that to look at things, both with elements and also, as you saw, these 3D models. It's been doing it. So the key thing is there that it's the magnets that's forcing the electrons to bend. But and basically, so, think about it as a big microscope that we can use to see all sorts of different things. And the good thing about it is that it's not destroying the object that we are looking at. So that's why we can use it for looking at paintings. That is incredibly valuable, for instance.
Because it just shoots light.
It shoots light through it, like you're doing, like an X-ray machine at when you go to the hospital.
But
but it's sort of a much powerful.
So it's like a light wave scope, microwave, light wave.
Well, microwaves are different. It's a different sort of wavelength of the. It's not the same type of radiation, but it's a.
But it's a microscope with a scope with a wave.
It's a microscope with a light, yes, where you see it with light. So it will be an X-ray. I'm not explaining this very well. I'm sorry, but it's
works.
And so you use that to kind of look through the forearm and understand its age in as that is determined by salinity levels. And
I'm using it both to see structures, like when I'm doing the tomography. Then I'm using it to look at how the shape and form of the forearms, the shell weight. I can model that, but I can also looking at the shell thickness and the more how it looks like in detail, both inside and outside. But then we can also use it to look at this elemental composition, like basically what kind of elements is in there. Can we see the manganese? Can we see the calcite? We can see, of course, but also strontium or barium or other elements. We can't see isotopes. We will not see differences in isotopes. We can't do that. But it's so we can make these maps where we're looking at, and we can make 3D with a microtomography. We're also making 3D images, of course. So that's what we have been doing a lot with students as well, to print in 3D images of forams. So that's I don't have anything at home, otherwise I would have shown you. It's very nice to get a big foram with all these very nice shapes and forms. But that is just, as I said, pretty pictures. So it's more interesting what they actually are saying.
But I have a question regarding the shape. And so when you have them big, they look like other shells, right? They're very similar in shape. And so I don't know if you can talk about why there is this correlation in the visual between these tiny forum.
You mean why nature has decided that that's a good shape to make, or
nature that decides?
Sorry?
Yeah, if that's nature's decision. It's just interesting to think about whether there's a function in all these different organisms.
I think it is. I think it is. It is how you can. I think there was a PhD thesis about this not recently, where they sort of mathematically describe this. That it is how for the forams how they are built is the most. I think it is the most how to build it more efficient. So to sort of save energy, how to do that, how to get the most connection with it. So all the chambers are connected for a hole inside them. And I think that the way they build them is the most energy efficient. So I think that there's definitely a relationship to how larger shells are also built with this sort of spiral form. But forams are so many different forms. I mean, it could be also, they can just be elongated. They can be biserial when they are just two rows of chambers next to each other like this. They can look like a cone. They can look like a cinnamon bun. They can sort of have all sort of shapes and forms.
But it's so nice. Sorry, I'm just going to say my reflection. It's so nice when we say how to get the most connection. That really sort of connects it. Thank you.
Desiree, I think you were the next.
Yeah, I was wondering if the spiral is important or isn't very good shape to build on or so, but is it also in the other, is there any kind of spiraling going on in the other shapes as well or so, maybe maybe in a pattern or some kind of.
Yes, we have these that we call unilocular, which is basically just one chamber after each other. That's sort of very simple. That's not so much of a spiral shape, but I think most of them would have an axis and have something. It's very common that they grow sort of in a spiral shape like that.
But because I was thinking with Josephine's question also, that also in the human body and movement, so maybe it has to do with movement as well. There is also circulation, and spiraling is always the way how you do things most easy or so. When you that's what I know, because I do a certain kind of gymnastics, what is about the chain of the movement. And maybe there is also so maybe in nature the spiral is like the most what did you say the word is?
Energy
economic?
efficient.
Way to do.
I think that I think that should be calculated by somebody. It should be quite easy to sort of see that. There was something, yes, and then of course the shapes from the planktonic ones, the ones that are living up in the water column, they tend to be quite bulbous, like popcorn, more or less, and that is because they need to float. And they need to float. Often they are adapted to which water depth they are living at. And then they are sort of adapted in their shape depending on where they are adjusted to live. So, but most of them look like popcorn. But the sort of shape of the chambers will be different compared to, well, depending on where they live in the water column, which is also rather fascinating. Sort of how do they sort of evolutionary I can see how they have done it, but it's still fascinating that they have. Cecile, I think you had a question.
Yeah, I now was just reminded of the 3-dimensional spider web that maybe we could see a picture.
Of course, yes. Yes. No.
May I ask another question while we're waiting for the picture?
Of course.
I wondered, you mentioned you could look back 600,000 years, and I wondered if that's a limit of some sort.
No, that was just that image. No, so as long as we can find forams, we can use it. It depends a little bit on the preservation of the shells and so on. But for isotope records, they have done it for the last, I think it is the last 5 million years or even longer. So it depends. But of course, when it's getting so old, then they are a part of the bedrock, or they make up the bedrock. So sometimes it can be tricky to get just the foram and not the material that surrounds it. Oh, now I have a problem with my mouse. So that was just that picture. So that wasn't. Let's see. So more questions.
I have kind of maybe a more general or big question, but at the end when you were talking about the specific data with the Baltic Sea and the changing levels of oxygen, and you know, that's one
Yes.
kind of data that we get from these organisms. But I'm just curious for you, what kind of stories forams tell, or what are some examples of stories that forams tell about, you know, the history of this planet that you think are most important to transform into another kind of data or another kind of communication? Like besides the Baltic Sea for the last 20 years, is there something that comes to mind for you that you feel like is important to communicate?
Well, I think that, well, we have done a recent study from Santa Barbara Basin where we show that forams that they have adaptations where they not only using oxygen for their cell respiration, but they're also using nitrogen, and that they perhaps can adapt when we have an environment with increasing low oxygen conditions. Maybe they will be a winner in that environment. But then I also think that in general, it's very easy when you talk to the public that when you talk about global climate change, and then you're talking about decreasing oxygen content due to increased warming, and you also talk about ocean acidification. People tend to be scared and upset, and just like, oh, there's no point in doing anything because everything is just going downhill anyway. So people getting climate anxiety, and then they think about plastic, and they think about eutrophication and pollution in general. And I think that both, I mean, maybe there's two parts of this, both showing that the environment is worth saving, you know, showing all the beauty of the world and the biodiversity, and showing that we actually need to preserve this. And why shouldn't we? I mean, who are we to say that which species should live and not live? So I mean, just by showing the nature's beauty, I think that or ecosystem in general will make people more keen to take actions to preserve it. But then also to say that we need to do something about this. And I think that this is where for lots of reasons, but that climate scientists in general have failed to communicate this message. And then, of course, there is politics and all this, but I think that ways where we can, not in a naive way, but in some way reach out and make people aware and interested and willing to change. I don't know if you understood me there, but there's sort of two questions in this, and I think that I can show, I can show graphs about oxygen concentration till the moon comes up or whatever you say. It doesn't change people's willingness to change. But maybe there's other ways to present the same message or another direction showing the same thing that also not perhaps causes anxiety and people just being shut off and say that, well, yeah, maybe that is the most important actually. Because I love data, but not the whole world loves data.
I have a question, and also I have actually two things. So at one point you said knowing the past, to understand the past you have to understand the present. And I always think that we have to know the past in order to
I think it's two. Yes,
present.
I think it's
So,
yes. I think it's two. I think that it's both of them. I think when we are understanding, when we are looking at the past, yes, we can understand the context we are living in and why we're doing the things that we are doing. But I also think that when we are looking at the geological record, we also need to understand how the present day is functioning, especially when it comes to sort of different marine settings. And that's why I'm pushing my students always to make these graphs. Is the temperature that we have reconstructed, is it realistic? And they don't understand that, they don't see that until they have looked at the numbers, how it looks today. How is the large variability today? Is this even reasonable to think that is going on? Now I promise to show you this picture of the pseudopodia.
Oh yeah. Then I will do my remark afterwards.
Yes. I also have a movie. I don't know if that would work. Let's just see. So here is a forum. You see my arrow here, what I'm trying to point out. And then you see this structure around them coming out of the pores of the forams. And this is what it's using for basically lots and lots of activities. And then here is another forum. It's completely round, and it has this network of pseudopodia putting out. And you see here it has caught a shrimp, basically at the same size as itself. Basically the same as a spider web. It caught it. And let me see if this will work. This probably will not be any sound to this, but it's let's see. It would just have to.
We still just see your presentation. I don't know.
You don't see. You don't see. Oh wait.
Reshare.
Sure.
What do you see now?
Your presentation.
Yeah. So you have to switch the
you have to
screens.
Oh, okay. Let's see. Reshare.
Yeah. Now we see it.
This was not the one that I wanted. Damn it. Do I have to do another reshare now?
Yep. Or if you can look it up in YouTube, but if you.
Yes, this is the one I want. And
So cool.
so, wait. Do you see a forum moving around now?
Yeah.
So it is full of little diatoms, that's phytoplankton, that it's scooping up with the pseudopodia. And then you also see how tiny these diatoms are.
Wow.
Okay. Oops. Oh. No, I don't want to share anymore.
We see you.
See me. I don't want to. I have a strange sound now in my ears.
Do you want to mute and unmute maybe?
So, okay. I'm back. I think I'm back. I don't look at YouTube anymore. Or we together. Yes, so you saw that this sort of spiderweb structure that it's doing. What else? You had another comment, Desiree, I think.
Yeah, that I think it's so amazing that a one cell species can build this shell, and also that it is like we think when on the beach, you think like a sand grain is a sand grain, but then there are also, those are also forms, and like those postcards also, like that it is under the microscope and so beautiful made, and then all those different kind of forms. And now I'm like, yeah, everything is movement, of course, and like we are all yeah, atoms or so that moves, and nothing is actually like, maybe, yeah, we can feel ourselves like a body or so, but then everything is movement. But this is for me like, this explains that, yeah, gives me more grip on that kind of feeling, because I love to think of that thought that actually everything is not like is not so, is more fluid than we think. And now, yeah, this also that this can be so small and so defined, actually, yeah, that makes it understandable, or so that there's a world in a world in a world that is always continuous, or so. That is so beautiful. That is what I wanted to share. Like, I'm very happy with everything what you said. Yeah, thank you so much.
Thank you so much. So you are based in the Netherlands, right?
Yes, I am.
Yes.
Yeah.
So, you know, if you're going out to the tidal flats, the really muddy ones, and then you just scrape off surface sort of muddy sediments, sort of a little bit finer than typical sand, just a little bit. And then you can use a coffee filter or some sort of filter, and sieve it. You know, you can just use the shower head, and then you will see tiny white fragments, so tiny things, and then you can and there you have the forams.
Cool.
Yeah.
Yeah.
And I'm pretty sure that you will find Elphidiums when you are looking in a tidal flat in Holland.
Ah, cool. I will certainly do that.
Yeah, it's you always have to have a little plastic bag when you're close to the beach, because you never know. You might want to take a sample.
I just was at the beach. It's like a pity that I didn't know.
Yeah, yes.
Because I have plastic bags with me, and I always pick up things. So yeah.
And then so in Okinawa, there you have so this island south of Japan, where you have the star-shelled beach as well, or the star sand. You can even buy them at the tourist office there. That is actually also forams, but they are huge. They are several millimeters big, and they called the star sand. That's also forams, but bigger ones.
Cool.
Okay,
Can I ask another question? Do you have it in you to talk a little bit longer?
sure.
I wonder about what you were talking about, how this feeling of this information needing to get out and how it's a lot about environmental, the future of everything. And I wonder if that's something that drove you to choose this path, or if it's more something that you have come to realize because of what you've learned.
No, it's what I've come to realize, because when I was a student, I thought it was basically only a lack of information and knowledge that was sort of hindering change. I thought that if everybody just knew, then it would change and we would make the adjustments that is needed. But I've understood later on that that's not the case. We have lots of different motivations for what we are doing, and change is always very hard. And we need to communicate in another way. No, I thought that it was just a lack of information, to say it simply. Or maybe I didn't care that much either. Maybe I just was interested in science in general and data.
So do you think because of what you've learned, you've come to care because you've seen such stark differences, or what has it been for you inside of this process?
So I think that, I mean, it's different parts, right? I mean, I want to learn just and know things just because it's interesting to know them and find out things, and then we have sort of also a responsibility to change things. I mean, it's quite obvious that we can't continue the way we are, what that we are doing, and that's more sort of an activist, and that is more perhaps me as a person, and not so much me as working at the university. And there's always this discussion going on: how much can you be an activist when you're also a researcher or a scientist?
That would be so interesting to hear more about, actually, the way you and your colleagues speak about this.
Yeah, it's I think that's lots of us are thinking about that and discussing and debating, but it's it's I don't know, it's also, I mean, if you're speaking more in general terms about different topics, coming to realize when you're getting older, it's also that in the beginning, when I started university, I thought that basically everybody was treated the same. And if I just work hard enough and be good at what I'm doing, there will be nobody tries to block me or have just because who I am or what I represent. But with time, you realize that is also not true. And that can make you a bit upset at times. But then you also realize that's, with time, that change doesn't happen just because, I mean, it's like there is no vaccine you can take against unconscious bias, for instance. It's not like, okay, now we are informed that this is going on, happening. Now we will all be cured. It's there all the time, for lots of different reasons, cultural and what we decide to do. And so now I'm going off on a tangent. I'm sorry about that, but it's
Can I ask you a question which is a little bit to follow on what you were just saying about this coming to this realization of how to communicate, or like maybe the futility of communicating these things with facts. Are you of the personal opinion that arts can have an impact on these types of conversations and be like a good medium to formulate or to even bring up the highlight conversation or the issues, or to bring it to people's awareness? Do you think art is like?
I think so. I think art, absolutely. Art speaks in another way. So I think that absolutely art can be perhaps with a mixture, so it can be another yeah, another path, another mean that might be much more efficient. If I didn't think that, then I don't think I would be here.
No, exactly.
No. Yeah.
I have another question. It's a term you mentioned, the last glacial maximum.
Sorry. Yes, so that was 23,000 to 21,000 years ago. That was when we had the largest extent of ice sheets. And so it's a very nice room that Josephine is having.
Thank you. This is my studio in Abbott, but there's a dump truck outside. So.
Yeah, so that's what when we had the largest extension of the last ice sheet. That is when we had 2 to 3 kilometers of ice on top of us here. And the ice was extending down to northern Germany.
From the ground and up.
Yeah. Yeah, it's a lot of ice.
That's a lot of ice.
Yeah, we are still compensating for that. You know, that's why we have still the land uplift. Not down in Skåne, but up where I live. And this is where Höga Kusten is rising 1 centimeter per year, because it's still compensating for the amount of ice that pushed down the land.
So is there a point when the compensation will be done with? And
yeah. Yeah. So I mean, that in Skåne that was reached 3,000 years ago, and here in Skåne also there we have a sea level rise because of warming, and you know we have. But here we are still so basically whole Sweden is sort of tilting a little bit.
And there is a point of ultimation, or
yeah. So there's a point where I don't remember now. Maybe Anna remembers when when we will have when it will stop.
Do you mean
when will the Höga Kusten stop to?
No, I don't have that.
Yeah.
Time estimate, but they are not affected by the sea level rise due to climate anyway.
Yeah.
So
and this is another problem that is rather interesting, because then we have this land uplift in the northern part of Sweden, on the coast there we have large fiber banks deposits that were deposited in the 1960s, and then it was deposited below where wave action could get hold of it. This was from paper mills and so on. They thought if we just dump it out there, it will sort of not affect humans. But now with the land uplift, it suddenly is getting into the zone where wave action actually can get at it. So now that has become also another problem, because you know it slowly rises up.
So it means the stuff they dumped is now
getting shallower and shallower.
Okay.
It's not getting up on land, but it's just that it's getting so shallow that when waves are hitting it, the wave energy will start to erode these fiber banks, where we have a lot of nasty things. So there's all sorts of interesting aspects to it.
I'm wondering about the cores that you're taking up. If you're looking at them the way we've been looking at the core from Vombsjön, that on the very bottom is the oldest material, and you can just sort of go with that logic, or if there's something. Yeah. So that's the is greater in the oceans, it's the same logic.
It's the same logic. That's what we want to, and then there can be, of course, bioturbation sort of organisms or things that has moved things, but that's not what we want. We want it to be. But then, of course, we also have marine deposits up on land that has been uplifted during, for instance, glaciations, and pushed up on land, and then it can be tilted and moved around in different ways. So, but the basic principle is the same as when you've been working with Kalle and Anna, that we take a sediment core, the deeper we go, the further back in time we are.
And so how do you know that you get this thing that you want? Is there a way to
Yeah,
see
we can, yes, we date them with different methods. And then we also use seismics to see how sort of the layering of the seafloor is and how it sort of, if it looks undisturbed and so on. So it's, but mostly it is with dating techniques that we use radiocarbon, for instance, that we know that we haven't had any age reversals or things like that.
So the core that we extracted in September of last year, Anna, Kalle, and I was just told a few months ago that there's a hiatus that we didn't know of before. So there was new information, or there was rather like there was a pause of information, if I understood it correctly. And I wonder if you have that too, and what you do.
It can be, yeah.
And how do you regard that sort of breaking of your logic?
Yeah, so I mean, we had a record where you can have that when it's just different depositional environment, and then something has changed, like that had caused erosion, have caused that the currents there have higher current speed and transport away material, and then we could have a hiatus, so we can have a time gap, or we can have a change in sedimentation rate. But you don't want that. I mean, you want this continuous sort of accumulation of sediment. And this is something I actually didn't talk about. So how much material that is being deposited on the seafloor varies super much with where you are. So if you are close to the coast, it's about 1 centimeter, can be, but typically perhaps, well, let's be a bit more realistic, 3 to 4 millimeters per year. That is sort of accumulates. And then you're going out to the deep ocean, and then you can have 3 to 4 millimeters per 1,000 years. So you see the amount of sediments, that how much time each sediment, how much time each 10 centimeters of sediments represents will vary very much where you are in the ocean. So if you're close to land, then you will have land runoff, you will have material coming out from rivers, you will have much more, you can have much higher productivity, and then you will have this 10 centimeters will represent a much shorter time. Whereas if you're out in the deep ocean, where basically the only thing that is accumulating is things that are coming down from the surface, that are being produced in surface and slowly accumulates down at the seafloor. And then these 10 centimeters will represent a much, much longer time. The time is not static, depending on where you are. Okay, I feel like I've been talking a lot now.
Thank you,
You have asked so many interesting and highly relevant questions, and have made me think as well. So I'm very happy about that.
We're so grateful, really. I have one last just reflection to share about the map images you showed of the Baltic Sea, and just looking at how the term hypoxia has been such a acutely scary thing that's been happening in the population of humans over the past year, and how the blackening area around Gotland, I think, was on the map, also looks like a pair of lungs. I just thought about the acute parallel to the past couple of years that's been going on in the world. Just thinking of how to talk about things and translate things, and what it means. Hypoxia in the sea also can be translated into how difficult it's become to breathe in different ways.
It's true.
Yeah. Thank you so much, Helena. This has been truly super interesting.
Thank you. Thank you for being so focused.
Thank you, Helena.
Yeah, thank you so much for your time and everything.
Okay.
Have a wonderful afternoon, everyone.
The same. So what's the program now? We are leaving, or we are so what's
the program is whoever needs to go gets to go, and I think for the rest it would be nice to just debrief and chat some. So whoever needs to leave, thank you for today, and whoever wants to stay and chat and debrief, please stay. Goodbye, Anna.
Bye-bye.
Okay, bye.
Bye, Helena.
Bye.
Thank you. I don't know if Sana is still here. Probably. Hi, everyone. Is Adrian awake?
Yes.
Yes,
Great.
yes. Wonderful.
I've been thinking about what am I going to do now? It's 6 a.m.
It's
And
either
you
now
are,
or
of
get
course,
outside.
awake.
The sun is going to rise in like 20 minutes, says my phone. So
And how far away is the ocean?
yeah.
I think you should go to the ocean and bring a coffee filter and a small plastic bag.
Mm-hmm.
Did you all take notes?
Yep.
Yeah.
It'd be really fascinating to compare notes or in some way talk about that. We might not have to do it now, but it would be great for this part of our group to actually have a meeting, I think, and look
Mm-hmm.
at what we have recorded.
Mm-hmm. It was very cool, right? Oh, it was amazing. It was really, really cool. Again.
Oh, did she leave? Yeah.
Yes. No poor connection here. Well, does anyone want to offer another reflection?
I was interested. There was this one part, I think, where she was saying, like, if you look, I didn't really completely understand what she was saying, but it's like if you look at the water all over the earth and you look at these forams, the forams from all over in different bodies of water have the same hydrographic data, or maybe I was getting confused, but there was like something where she was saying that all over. Did anyone get that in kind of the beginning?
No.
No. Okay.
We should go back to the recording. I'm very curious.
Yeah.
I thought it was really fascinating when she talked about this passive sort of forced recording and how she's, on the one hand, really fascinating in these organisms, and on the other hand, she's using bleach to remove them from their recordings. How there is just this, these two parts where their lives are what sort of brings the recordings about, and how it's fascinating to try to look at different ways to, or environments for them to live in and what you can do. And then by removing them, we can see what they have recorded. I mean, it's just normal science behavior. I think there's so much charged language inside of it.
Yeah, I've been kind of thinking about the lake also, and then these forams as being like these kind of arbiters or like carriers or safe keepers of like records or data in nature, how they kind of like hold them and like keep them kind of static in some way, like they're like carriers.
Yeah, and then they're also just creatures of divine geometry, and they're made out of one single cell, but they can do more than we know how to do. It's just sort of mind-boggling to look at and compare oneself to.
It was kind of funny for me because I didn't get to sit through it the way you got to sit through it. So I think you experienced so much information and science, and I had these pockets of both visuals and little snippets of language and question or formulations of questions, and then like these other very bodily preoccupations with uni. And so for me, I think it's been super different experience than for you, because I was seeing you on the screen, but not always kind of hearing the conversation, because it's hard to multitask in this way. But I was screen grabbing when she showed this and it's funny because now I don't even really know what it is, but this organism which has this purple lining that build its own segments inside, and it could be because I'm structuring like choreography right now, but all I could think about was dance and costume. And then she was talking about these, I have like this really weird notes now, which one is of like shoots light through it. So now I have this little drawing here of this kind of dynamo winding up into like a twirling dancer wearing like organza with light shooting through it, kind of like a weird body but slash kaleidoscope with like particles moving and dust, and then like Desiree being so like joyous and happy to hear these things. I don't know, for me it was like it was really this kaleidoscope. It was like every single time I came in and out, there was this different fragment of things that now are very like, yeah, it's like constituted in these weird notes that are totally not probably what you heard whatsoever, because I wasn't really able to be part in that way. Yeah, I don't know. That's why I have to say.
But what you're describing ties right back into what Desiree was pointing out, which is this circular movement, and then how Helena was saying it's the most efficient, energy efficient way to do anything. And there's that's this way, and how there's on the one hand the point of energy efficiency, and on the other hand there's the point of as much connection as possible, and how they tie together in a spiraling movement.
Yeah.
And sort of some kind of divine geometry to that too.
Yeah.
And then I think when you're describing your kaleidoscope, it makes me think of the the organism that uses whatever is around it to create its shell or its meaning.
Yeah.
I think it's really, really nice that you're bringing it to choreography too, because what we want to do this year in this project, even though it's like wide open and we're just like collecting parts, it's like we're sitting here, we're each an organism sitting here, and things are sort of flying by, or we place ourselves within this environment, and then we want to construct something, but we also really, what we've been talking about doing is choreography, which is movement and touch. So thinking of connecting touch and movement, and just connecting us and this. I think that's the basis that we will move forward.
I mean, from these note takings, I already have something I want to make, which is quite funny, because I didn't think at all about this meeting as something that would, I just didn't think of it as something that would generate these kind of thoughts. I thought of it something that would be super interesting, but not necessarily something that would culminate in an idea. But I think because I got these fractions, it's like nonsensical, but totally logical for me to start thinking in this way. So for me, it was really, really quite giving. I'm not sure I want to watch the whole thing, because then maybe it becomes too, I don't know, might be lighter and not so bogged down with theory or like information. It's also quite nice to have this abstract associative elements.
Yeah, I would save it for some other time. But I would still love for us to have another meeting quite soon, where we maybe show each other our notes, or in some way look at our notes, and then also try to do something together. Be it movement or something else, but where we try to use what we just got and compare or combine in some way. So I hope everyone is up for that.
Yes, for sure.
Just a bit of experimentation. Cool. So let's just in some way or another set a date for that.
Yay, we are starting.
Plan it. Do you want to plan it now, or do you want us to do emails?
Why? Maybe that's a good idea, right? Or to say it like so that we more or less knowing what we're thinking of.
But are we having another lecture with Kalle next week?
I hope so. I haven't heard back from him yet. which I think is positive. But
Yeah.
I think he's also really busy.
Wasn't it? I think he said the 14th.
Yeah, I bumped into him on the lake. We were both on stand-up paddleboards the other night.
Nice.
But I fell into the water.
Of course. That's what you do.
But we didn't speak about this because I got wet. So I will ask him. But so possibly on the 14th. But I know not everybody who's here could be there, right? Was it you?
I'm not there.
Okay.
I have to work at the
Okay.
academy, so I cannot.
Do you know other? I mean, I can check. I don't know about the rest of you. I know Sanna is very busy and moving about.
Yeah, but yeah, it's a bit tricky right now because Samuel doesn't know which days he works, and then we are not sure when we go to Berlin and all these things. So, I mean, I'm gonna try and be there no matter what the date, but I can't 100%. So.
But the 14th might be possible for you.
Yeah. Yeah.
And Rebecca, I don't remember what you said about the 14th.
Yeah, I can do it on the 14th.
Great.
Me too.
And Adrian, are you still going to be with us?
I want to be, but I'm going to be traveling on the 14th. So I wouldn't be able to make that day.
Depending, would it also be at 1? Actually, I can't at 1.
Okay, so when can you?
I can before 1.
He teaches before. His window is between 1 and 4.
Yeah, dang. It's just. Did we say, was it the 4? Because I'm teaching Tuesdays from 2 to 4. So I can't that day.
I mean, I can talk to him. We'll see if he has another possible day. We have Anna and Kalle who both are going to do, I think, each a presentation. And she had more time moving forward, but I think the thing with Kalle and Helena is that they're both getting very, very busy at the university. So we will see, but it might be that you'll have to just be there for the first hour then, or for the recording. But talking about a meeting just between us who are not scientists, when is a good time and a good day? Let's look at this week and next week. I can do almost whenever. I can't do tomorrow.
I can do Sunday, like evening, or next week, pretty much every day except the time with a lecture on Tuesday, and the same time on Wednesday I also can't, but the rest is free. But it's a little bit like hard to remember, I guess, when I just say it like this.
All right. Well,
that's true. Just thinking that. Yeah, right?
Yeah.
Well, if anybody knows they're just unable or busy, that's a good way to begin.
I can also do Sundays. The only time I can do this week. I mean, I could do other times, but I just don't think it would be helpful because I would be so preoccupied with Uni that it just doesn't make sense. Whereas Sunday, Samuel's here also, so there's no problem. And then next week I could do, the beginning, Monday or Tuesday. Then it becomes increasingly harder in the rest of the week, but it also something I could maybe find the time for, but it would be beneficial for me to have it at the beginning of the week.
So Sunday night for the others, how does that sound?
I can't on Sunday, this Sunday.
Right. How about Monday?
Monday would be great. I would love Monday.
I also love Monday.
Okay.
Rebecca, do you love Monday?
I'm on a train on Monday, but Can you do it on Monday evening?
Because then it also works for Adrian.
Yes, that would be great.
I'm not there on Monday evening. I have to say, I'm sorry.
Mm.
Like not like from 20 to until 9:30. I'm not there. So
Would you be able to do earlier or later? How do we feel about sort of like a late night on Monday?
Depends on how late. I also have problems with too late. I can
I
do any
also
time.
remember from last time that these very late meetings we were tired.
Yeah,
that's also true.
Or something. I don't think it's the best.
What about like 7 or something like that, or 6:30
Yeah.
on Monday?
when
I can
does
do after 7 or earlier because I put her to bed at 7.
so Rebecca, when does your train
Or like-
arrive?
I think, yeah, my train leaves at 4 and I will be on the train for like 3 hours at least.
Okay.
So,
7.
7 or 8, I will be home, but maybe that's too late.
This is tricky.
Yeah.
But Daisy, do you have to be somewhere at 8, or would you be able to
Yes,
be
I have to be
8?
somewhere at 8. So I was just thinking that I need also time to go there.
Okay.
Could we
Like
do
half an
earlier?
hour or so. So maybe earlier would work. Yeah.
Like before, Rebecca, you said your train is at 4.
Yeah.
Could we do earlier in the day, like more around lunchtime or something?
I can't.
Okay.
So then the question is whether it would be possible at all for you to do it when you're on the train, or if that's just chaotic. Well,
On the train?
maybe later, either one of us is on a train, or we do it later in the week, because sooner than Sunday was not an option for some of you, right?
I can't.
Okay.
It would be difficult for me also. I would be with Uni basically the whole time. So
All
for
right.
the sake of being actually in the meeting, it would be super difficult.
I'm so glad we're not doing this by email, I just want to say.
Yeah.
It's nice. Yeah. Okay.
Well, I forget what you guys said, moving on from Monday. Was Tuesday an option?
For me,
Monday
it's
we
not.
can do the option, but maybe not 2 things on Tuesday if we're gonna have the other lecture as well.
Right, okay.
That becomes like a full day.
And so Daisy,
But
you're
if that lecture doesn't happen on Tuesday, then definitely Tuesday.
Yeah, if the lecture doesn't happen on Tuesday, we can do Tuesday after, like Tuesday evening, maybe.
How is that for you, Daisy?
So I work, I think until 5 or so, and then I have to travel back home 1.5 hours, something like that.
So we would be on by 8 o'clock, maybe.
Something, yeah.
Is that an option for you guys, even if we do have a lecture, or is that too much?
For me, I can't do that day, but you know, you don't need to.
You can't.
And there's also Wednesday and Friday, like that are my days that might available. Oh yeah, Thursday as well is like more available for me. I don't know. I just saying my things, then I
Thursday,
bring
Friday would be
something in.
great, 17th.
Yeah.
Works for me.
Are we looking at
Thursday is my birthday, so
Your birthday.
so a party? Maybe
I
that's
think
not.
yeah, there are plans for us to go out into nature. So I think I'm not gonna be very comfortable.
I have a radical suggestion, which would be tonight.
That's not possible for me because Samuel doesn't come home until late, so I have to do all the bedtime, everything on my own, and then yeah, it would be very. I'm fine with if you want. If it's the only time all of you can do it, I of course totally understand. You should go ahead, but I don't think I will be very present.
And after she goes to bed, it's too late for you.
Sorry?
after she's asleep is too late for you?
Yeah, it's just whether I'm gonna be totally present because she's
Right.
not a great sleeper, so I might be up and
Right.
up and out. But it's possibility. It's just that I'm not gonna be like 100% guaranteed that I'll be fully engaged in the thing, because I might just suddenly not be here and then not be here for the rest of the meeting. It happens quite a bit. So of course, ideally, I would love to be able to be part of it, like with certainty. But if the only time that works for everyone else, I'm totally willing to try it.
All right, so we have a maybe option of tonight, but then we would also look at Wednesday, Thursday, or Friday next week. But then Thursday is not an option because that's your birthday.
Okay.
All right. Is it the 17th? Wasn't? Let's have to look at it.
Friday is the 17th.
Oh, okay.
Can do any of those days, Wednesday and Friday, and also next weekend, which is not an issue. It's
I
just
can't do it.
basically. I can do all week next week with the except for Thursday.
I can't do
Monday?
Wednesday, but I could do Friday, Saturday, Sunday.
Yeah, I can also do Friday, Saturday, Sunday.
Donna, did you say you can or you can't on those days?
Who are you asking?
Sorry, I lost the connection. I was asking, did you have availability on any of those dates, Sanna?
Yeah, I can do any day except for Thursday.
Oh, okay.
Yeah.
So Friday,
Next
Saturday, and
week.
Sunday are.
Oh,
Yay!
it's fine.
Maybe.
How was crazy?
Cecily?
It's just this week I can't do it, but next week I can do it. Yeah.
Friday, Saturday, and Sunday next week.
Yeah.
I have time.
So,
17th, 18th.
so should we maybe do it then on Friday? Is
Yeah.
that a good day? Because everyone can.
Yeah.
That's really cool.
Yeah.
And it's maybe we talked in beginning of the year, you know, and a little bit about last project, and then we said maybe we should have the same day every week or every other week or whatever, that we have a time set, and so that we not always have to search for a date.
Yeah.
Because there are enough things like events, like with a geologist, and so that we have to search for a date. But maybe for our process, now we started, maybe we can have that, because yeah, I do a lot of things, and then yeah, and I have to be careful with my energy, and maybe that's handy.
Sounds
And
great.
I thought, right, okay, cool, because we said that we thought it would be nice, like Cecily, I think.
Yeah, I think
Cool.
I'm up for that, and then sometimes we might have to rearrange.
Yeah,
things.
sometimes.
Generally,
Yeah,
I think
sometimes.
absolutely. If that works for everyone, I'd be very happy to.
Yeah, and it can also be like maybe we have this meeting with all of us, and maybe some cannot join, or so like someone cannot join because of something else. Okay, maybe that can also happen. Or sometimes we maybe have to switch, but then we have a sort of regular.
Yeah, sounds great.
Good.
So time on Friday the 17th.
So cool.
Adrian's time difference is 9 hours, so we want to be mindful of a 9-hour difference. How early is the earliest you want to get up? Because 4 a.m.
I could do 7 a.m.
It'll be 7 plus
my time, so that's
4 o'clock for us in the afternoon.
Okay, 4 o'clock. Good.
How's that for everyone?
For me, it's good.
4 o'clock.
Okay. So 4 o'clock on Friday the 17th. Great.
That's my
nice.
3 o'clock.
Yeah, that's your 3 o'clock.
Yeah, that's fine.
Wonderful.
I have to run, honeys.
Okay.
So I'll see you then. I kind of hope that the lecture won't be on Tuesday, but I don't know. We'll see.
Me too, of course. But yeah, if it is, then it is, right?
Yeah.
But I really hope to be there, actually. Also, it's Kalle. So he will have so many things that are interesting.
Yeah.
Well, yeah, always when he tells something, I'm like, you know, I want to know more. So I'm like,
yeah, exactly.
and then I'm not there. Yeah.
It sucks. Yeah.
Well,
but, we'll see.
thank you so much, everyone. Like Josephine, to start the process of like this. Like, yeah, we started already up, but now we are, in our process together. It's really cool. Thanks.
Thank you so much. I
Yay.
appreciate all of you and your patience and your showing up.
This is really wonderful. The lecture. Yeah. Yeah. So thank you.
Hi.
Bye bye, Cecily.
Bye.
Have a lovely day.
You too.
I'm going to end this meeting unless anybody has anything to add. All right.
So have a beautiful day, everyone. Bye-bye.
You guys have a good day. Nice to see everyone.
Yes, for sure. For real.